So I hope everybody is doing well. Welcome to my channel if you're new, and if not, then welcome back. So today we are actually going to deal with carbohydrate metabolism. Now, we were actually dealing with protein metabolism as such, but I'm not actually making these discussions in a particular set of order. So I'm going with, whatever catches the flow. And hence, today we start with carbohydrate metabolism. So we were last studying about the amino acid metabolism, at least in my last video. And we ended with the general metabolism of amino acids. So the general metabolism of amino acids, we completed that with our last video, with the urea cycle part. So we would have started the specific amino acid metabolism and talked about the different amino acids individually as far as their metabolism is concerned. But I think that carbohydrate metabolism, or rather glucose metabolism as such, it's a more fundamental thing to know. So we skipped over to here. So today we are going to talk a little bit about the metabolic pathways of glucose. So now here's the thing. In the entire biochemistry, whenever we'll be studying about any metabolic pathway, or rather any series of metabolic pathways pertaining to a particular set of compounds, for example carbohydrates, fats, proteins, nucleotides, etc., we always go by the order of, first we talk about the digestion of that particular thing in our digestive tract, the absorption, then we talk about the actual metabolism, okay, the actual utilization of that substance by our body, which actually pertains to metabolism. So about carbohydrates also, we'll be following this very particular rule. So we start with the digestion of carbohydrates, then we move on to absorption, and then we move on to the different metabolic aspects of carbohydrate metabolism. Okay, so today we are talking about carbohydrate metabolism. So now, as far as the digestion of carbohydrates is concerned, I think we have all studied the digestion part many times. In many of our previous books, we have studied this in general biology. So there's nothing much to talk about the digestion of carbohydrates. Like we already know that the process of digestion of carbohydrates starts in the mouth itself by the salivary alpha amylase enzyme. We all know this, right? So the salivary alpha amylase, what that would do is, it would actually convert the starch or the boiled starch in our diet to maltose. Now the entire 100% of the starch is obviously not converted. The digestion is partial, about 30% or so is done. So that's why I'm not actually stressing much upon the digestion of carbohydrates part. Okay, so do me a favor and please go through it like you did when we were talking about amino acid digestion or protein digestion actually. So there's nothing much to talk about it. So please go through it yourselves. It's fairly easy and we have all talked about it a lot of times. I want to dedicate more time to the more important things to discuss about. But one thing I'll tell about the digestion part that is pertaining to the clinical application of digestion of carbohydrates, that is important. So let's talk about the clinical or the applied part of the digestion. Okay, so clinical application of digestion of carbohydrates. For that, we talk about a very, very common condition which is known as lactose intolerance. Lactose intolerance. I think everybody has heard about this very condition. So what is lactose intolerance? In the name itself, the meaning is hidden. Lactose intolerance. So intolerant to lactose. Now lactose itself, we know that it's a disaccharide, right? Made up of galactose and glucose. Anyways, so what happens is that this lactose in our diet, when we consume lactose and when it actually reaches the intestine, the brush border epithelium of the intestine actually has this special enzyme which is known as lactase. Okay, lactase is an enzyme, right? This lactase enzyme, what it would do is break down the lactose into galactose and glucose, and hence this is the part of the digestion of lactose. Why? Because we all know that lactose is a disaccharide, right? And disaccharides as such are not absorbable. The absorbable forms of carbohydrates are the monosaccharides. So ultimately the monosaccharides have to be formed, right? So lactose is converted to galactose and glucose by this enzyme lactase, but in patients with this condition, lactose intolerance, what happens is that very, very obviously this enzyme is absent. Lactase is absent, or it may also be deficient. Okay, so if lactase is absent, then there is no enzyme to hydrolyze lactose, right? So what would happen is that this lactose, it will start accumulating. It will start accumulating in the intestine, and that would in turn lead to conditions like I can say diarrhea or flatulence, right? Diarrhea, flatulence, these are some of the manifestations of lactose intolerance, right? Right. So this condition, it actually, how can it be treated? It can be treated by removing lactose sources from diet. So we should not consume, if we suffer from lactose intolerance, we should not consume food which actually has lactose. For example, milk. We should not consume that. Why? There is another reason for why we should not consume it. See, the implication here is that, listen to me very carefully. This enzyme lactase, it is actually an inducible enzyme. Inducible enzyme. So it is induced by its substrate, right? So if lactose is absent from diet, lactase being an inducible enzyme, so if it is withdrawn, the lactose sources are withdrawn, then these conditions, they will also go down, right? So if the lactose is not present, then obviously these conditions due to lactose intolerance, they will also not be present. So the best treatment is obviously to completely like stop any kind of lactose source, okay? And we can also start consuming curd. Instead of milk, we start consuming curd, and the reason for that is that curd has a lot of lactobacilli. Lactobacilli. It has a lot of these bacteria which actually have lactase enzyme. So curd has ready-made lactase in it. So we do not need to go through the pain of having lactase in our body itself to ferment curd. Curd itself has it. So patients with lactose intolerance can in fact have curd. And by the way, the cause for lactose intolerance, it can be something like congenital cause, so present from birth, or it may also be acquired. Right? So that's just a small thing to remember. Okay. And another thing is that in case of the treatment of lactose intolerance, I said that A, withdraw any kind of lactose source from diet. B, consume alternatives like curd. And C, use things like yeast, because yeast also has abundant lactase. So yeast can also be used for treating symptoms of lactose intolerance. Okay, done. So that's the clinical application of digestion of carbohydrates. The rest of the actual process of digestion of carbohydrates is very simple, and we have all studied it a lot of times. And so I think that the better thing would be to move on to more pressing matters. So we move on to the next thing on our list, which is absorption of carbohydrates. This is important. So let me use the blank space here. No meaning of wasting pages. So absorption of carbohydrates. Let's talk about absorption of carbohydrates for a bit. So see, I have already said that by the time the digestion ends, monosaccharides are the only form of carbohydrates which will remain, or at least which can be absorbed. So absorption of carbohydrates is only true for monosaccharides. Okay. Now, where are the monosaccharides absorbed? Are they absorbed in the mouth? No. Are they absorbed in stomach? No. They are absorbed in the intestine. Okay. They are absorbed in intestine. Now we know that monosaccharides, there are 3 monosaccharides for carbohydrates. What are they? They are glucose, fructose, and galactose. Right? Now, an interesting thing is that absorption rates for these 3 monosaccharides are different. So these 3 monosaccharides are absorbed at different rates. For example, galactose is absorbed at the highest rate. Glucose is the second, and fructose is the least, or rather the slowest in case of absorption. Okay, so galactose first, glucose second, fructose third. Done. Now let's talk about, since we are specifically talking about glucose as such in our discussion here, because it is the most important. It's not that we are biased or anything, it's just that glucose is the most important form. So let's talk about absorption of glucose in a little bit more detail. Now what's seen is that this glucose, it has specific transporters. Now transporters, these transporters, what are they? They are nothing but very special transmembrane proteins. We talk about transporters and the different transmembrane proteins, integral proteins, peripheral proteins, when we talk about the cell, right? But we do have a fundamental understanding of these things, right? So transporters, as far as glucose is concerned, glucose has these very specific transmembrane proteins which will transport glucose from the intestinal lumen to the inside of the intestinal cells. That is the job of these transporters, right? There are a variety of transporters for glucose. All of them together, they are known as GLUT. That is glucose transporter. As simple as that, right? So together they are known as GLUT. So there are different kinds of these GLUT. There is GLUT 1, 2, 3, 4, 5 and so on, right? But GLUT 1 to GLUT 5, they are the most important. Okay, so what I am talking about, let me repeat it again. Glucose has these very specific transporters or transmembrane proteins which will transport the glucose from the intestinal lumen to the insides of the cell, and these transporters collectively are known as GLUT, abbreviated as GLUT, and known as glucose transporters. Okay, as simple as that. Now, apart from GLUT, that is glucose transporter, there is another special type of transporter that is known as sodium dependent glucose transporter, or if abbreviated, S-GLUT. So we have GLUT, which are the normal things, normal glucose transporters, and we also have these special sodium dependent glucose transporters, or S-GLUT. What are these S-GLUT? What are these GLUT? What is the thing behind them? Let's talk about that. Okay. So what's seen is that, see, let's talk about the S-GLUT first. Sodium dependent glucose transporter. Now, when we study about the cell, when we study about the different transport mechanisms across the cell, we come across a very special kind of transport mechanism which is known as co-transport, right? Co-transport. Or we also come across active transport, passive transport, facilitated transport. We come across these terms. So S-GLUT, this transporter, this is actually a co-transport system. Okay, it's a co-transport system. Co-transport means there will be two things or two different substances which will be carried across by this specific transmembrane protein, and hence it's known as co-transport. We'll talk about the co-transport systems in details when we talk about the cell. So what are the two things that this S-GLUT transports? One is obviously glucose, right? And the other is also obviously sodium, because it's there in the name itself, sodium dependent glucose transport. So glucose and sodium, both of these are actually transported by S-GLUT, right? So this is actually an example of a secondary active transport. What is a secondary active transport? Again, we'll talk about these all things in details when we talk about the cell, the biochemical perspectives of cell and transport mechanisms. We'll talk about this. But just to understand a little bit, the fundamental at least, see, we all know that active transport means when something is being transported against its concentration gradient and hence utilizing energy. That is active transport. We all know that. But active transport itself can be primary active transport or secondary active transport. Primary active transport is when the energy which is used for the particular substance to be transported against its concentration gradient, when that energy is utilized directly, okay, so there is some kind of ATP hydrolysis or something, nothing else is linked. Are you getting what I'm talking about? Primary active transport means only that substance. For example, if we are talking about for example, hypothetically speaking, the primary active transport of a particular substance X, then when X is transported against its concentration gradient, ATP hydrolysis takes place, energy is utilized, and X transports against its concentration gradient. That is known as primary active transport. But here, this sodium glucose transporter, the sodium dependent glucose transporter, S-GLUT, this follows a secondary active transport system, which means that here glucose is being transported against its concentration gradient. So energy is utilized. Energy has to be utilized, right? But that energy is not directly coming from any energy source like ATP. Rather, it is being linked with the transport of another substance, which is sodium in this case. It is linked to sodium transport. Now, listen to me carefully. Sodium's concentration gradient is not against but for. That means that sodium, the sodium is moving from outside the cell to inside the cell. But that moving is not against concentration gradient. That is down the concentration gradient. So for sodium, the transport is passive. It does not need any energy. But glucose needs energy. Right? Now see how glucose transportation is linked with sodium's transportation. See, once the sodium reaches, okay, let me draw a diagram very quickly. Suppose this is the cell, okay, and obviously this is the cell exterior, right? Suppose this here is the S-GLUT. So S-GLUT will have two receptors, right? One will be for sodium and one will be for glucose. See, glucose concentration outside the cell is very high. Inside the cell is low, right? So glucose is being transported against its concentration gradient. Sodium, on the other hand, is a little low outside but higher inside, right? So, or rather, oh, I'm so sorry, I actually jumbled it up. I'm very sorry. Just switch these things. I said that the glucose concentration outside is high. No, the glucose concentration outside is low. Very sorry, my bad. Glucose concentration outside is very low, but glucose concentration inside is quite high. Whereas sodium concentration outside is quite high, that inside is very low. Okay, so I hope you understand. So what will happen is that the cell will use the sodium glucose transporter to actively carry glucose inside the cell and passively carry sodium inside the cell. But there is another transporter, and this is a famous transporter. This is the sodium potassium ATPase pump. And what this pump will do is, this will pump out this sodium back to the extracellular compartment. So this pump, sodium potassium ATPase pump, this will utilize ATP. This will utilize ATP, hydrolyze ATP. This will require energy, because now you're carrying sodium from inside the cell to outside the cell, which is against sodium's concentration gradient. Okay, so see, that is the reason how this ATP, which is not directly linked with this, it is directly linked with sodium potassium ATPase, but indirectly linked to the sodium glucose transporter. This is an example of secondary active transport. Okay, so the energy is not directly utilized as such from any source. It is rather indirectly utilized from a different transport mechanism. Hence, the sodium transportation is important. Hence, it is very, very important. And see, there is a very important clinical or applied part also to this sodium glucose transporter. So that's why I am talking so much about the sodium glucose transporter. If you do not understand this, it will be very difficult to understand the clinical implication of sodium glucose transporter and how we use it in a clinical practice. The clinical part of the sodium glucose co-transport system is that we use this oral rehydration solution, ORS, right? We do use this ORS, right? So this ORS works on the principle of sodium glucose co-transport. How? This ORS will contain both salt and sugar solution. Okay, so it's both sodium and glucose. So what will happen is that ORS is given in cases of severe dehydration or severe dehydration of body. That severe dehydration might be due to cases like diarrhea or excessive sweating, right? Like you are going out on a sunny day and you have a tough day ahead outdoors. So often ORS is recommended in situations like that. So what happens is that due to severe dehydration, sodium concentration of the body goes down, which is not at all desirable because it's very, I'm even tempted to say fatal, because sodium is that important in our body. So what is actually ORS doing is that it contains both sodium and glucose. Now what our body will try to do is that it will obviously try to absorb glucose. Because glucose is that important for the body. And since glucose is co-transported with sodium, so sodium is also absorbed by the cells. Okay, so co-transport of glucose and sodium by the S-glut system is utilized by ORS, and hence the sodium concentration of the body is restored. So it's an important thing. Okay, I hope you understood this. Now, S-glut is obviously a transmembrane protein and it's a transporter, but it has two forms. So there are two kinds of S-glut. One sodium dependent glucose transporter is known as S-glut 1. Yeah, it is known as S-glut 1, and the other is known as S-glut 2. Okay. Now, S-glut 1 is found in the intestine, whereas S-glut 2 is found in the kidneys. Okay, and both of these have their own clinical implication, but we are not going into that much detail. We may talk about this later, but for now this will suffice. Okay. So we talked about S-glut. That's one kind of transport mechanism for glucose. Now let's quickly talk about the other glut. We talked about S-glut. Now let's talk about the glut, the glucose transporters themselves, which are not dependent on sodium. Okay. So, glut, as I was talking about, they have different, it's an entire family of transporters. So they have different members. So GLUT1 is the most common glucose transporter, and it is actually uptaking glucose. It actually uptakes glucose from almost all cells. Okay, so for the most cells, GLUT1 is the transporter protein present, which will actually help in glucose absorption. Okay, GLUT2 on the other hand is a bit special. See, GLUT2 is very important. Why? Because GLUT2 actually is used as what we call to be the glucose sensor in beta cells of pancreas. The glucose sensor in beta cells of pancreas, very, very important. Now do give me a patient hearing. See, GLUT2, this is that kind of a glucose transporter which actually the cell is using for what we call to be uniport facilitated diffusion. Okay, uniport facilitated diffusion. So GLUT2 is following the uniport facilitated diffusion path or system, just like as GLUT was following the co-transport secondary active kind of transport mechanism. GLUT2 is uniport, so only one kind of molecule or one kind of substance is transported, and that is obviously glucose. And facilitated diffusion. So diffusion, we all know, is a passive process. Facilitated means that it is the diffusion which is passive is being facilitated by a particular transmembrane protein. In this case, it is GLUT2. So see, GLUT2, what we call, it has a high Km value for glucose. Now see, this is how GLUT2 is special. Just remember this. GLUT2 has a very high Km value for glucose, which means that if we do have obviously the prerequisite knowledge of enzymes and how enzymes work, we would know that a high Km value for an enzyme, a particular protein, would mean that it has a low affinity, right? It has a low affinity for glucose. A high Km value for glucose means a lower affinity for glucose, right? So since GLUT2 does not have that much of an affinity for glucose, it's not used as such as an extensive transporter. It is used as a transporter, it is used obviously, but not as extensive as GLUT1 was used. Rather, GLUT2's usage is in the form of glucose sensing, because since it has a high Km value, so it can sense the presence of glucose much better. It can sense the presence of glucose in the bloodstream much better. So if GLUT2 senses a high glucose, it will trigger insulin. It will trigger the beta cells of pancreas to release insulin. Okay, so GLUT2 is very important in that case. Okay, Done? So in this way, with the help of GLUT2, the pancreas is able to monitor the glucose level in the blood stream. Okay? And obviously the rate of insulin secretion is actually affected by GLUT2. Done? Done. And by the way, another very important thing is that GLUT2 is present in the liver. Okay? This is also an important thing. Just remember this for me, please. Just like GLUT1 was present in most cells, GLUT2 is mostly present in liver, the hepatocytes. Done? Done. Now, as far as the other glucose transporters are concerned, there is nothing much to talk about them. Like, they all have different characteristics, but GLUT1 and GLUT2 are the most important. And along with GLUT1 and GLUT2, there is another one glucose transporter, which is GLUT4. These are the three glucose transporters which we should know a little bit more individually about, because they are a little bit unique. So glucose transporter 4, it is actually, why we are talking about this is that it is the major glucose transporter. It is a major glucose transporter in the skeletal muscle and adipose tissue. Okay, so this is the first uniqueness of GLUT4. It is that it is the major glucose transporter in the skeletal muscle and adipose tissue. Now, when I say skeletal muscle and adipose tissue, immediately something should strike you. These two sources, these two sites, they are also known for another thing related to carbohydrates. What is that? Yes, very good. Both of these are sites of storage of carbohydrates or glucose in the form of glycogen. Right? Adipose tissue also stores, skeletal muscles also store them. Right? Both of them are actually sites of storage of carbohydrates. So, now this is how the next information will be connected to this. The next information about GLUT4 is that it is under the direct influence of insulin. Very, very important. It is under the direct influence of insulin. No other, and I repeat, no other glucose transporter are under the control of insulin. So this is also a very special information. No other glucose transporter other than GLUT4 is under the direct influence of insulin. Okay, so that's why it's very, very important to know about this. Okay, so what insulin will do is that insulin will actually induce the what we call to be movement of intracellular GLUT4 molecules from within the cell to the cell surface. This is what insulin will do. So insulin itself will be released when glucose concentration is very high. How insulin will tackle that is that it will, or rather one of the ways in which it will tackle that is, it will move on, the hormone will move on to these target tissues, which is the skeletal muscle and adipose tissue, and it will actually induce the movement of intracellular GLUT4 molecules. So the cell, suppose this is the cell, and the GLUT4 molecules, they are within the cell at the beginning. Okay, within the cell, not at the cell membrane. But when insulin comes, it will induce it. It will induce these GLUT4 transporters, and so as a result of that, what will happen is that these will move on to the cell surface. Okay, so now if they move on to the cell surface, they are glucose transporters. So what they will do, they will actually lead to binding of glucose. They will lead to binding of glucose and will transport the glucose inside the cell. Okay, so the extracellular glucose concentration will go down, right? So glucose will be stored in the form of glycogen. Understood? That is how GLUT4 will work under the control of insulin. Okay, so glucose concentration outside the cell will decrease, obviously, right? Now in diabetes mellitus, what happens is that diabetes mellitus, obviously it has different types. I'm talking about type 2. In type 2 diabetes mellitus, what happens is that these GLUT4 molecules, type 2, let me write it, these GLUT4 molecule concentration decreases. So there is not much GLUT4 molecules left for the intracellular transportation of the glucose from outside the cell to inside the cell, and hence store the glucose in the form of glycogen. Hence the glucose concentration in the bloodstream increases, or rather keeps on increasing. That is diabetes mellitus type 2. Okay. So and obviously there are other glucose transporters also. For example, if I just give one more example, obviously they all follow the number GLUT1, 2, 3, 4, 5, 6, 7, like that. Up to 10 also there is glucose transporters. But the thing is that just one more information I would like to give is that GLUT5 is considered not as a glucose transporter but as a fructose transporter. This is another important information. It is a fructose transporter. Not that it does not transport glucose at all, but it's a very poor transporter of glucose. Fructose is the main thing that it transports. Okay, done. So with this we actually or rather, okay, one more thing. We talked about the glucose absorption. We also kind of am talking about the fructose transportation also. So what about galactose? Galactose is also transported along with glucose by the same transporter, which is S-GLUT. Okay, so S-GLUT not only transports glucose, but it is also used for the transportation of galactose. Same energy dependent process. Okay, same. Done. So with this we kind of finish off with the absorption of glucose. So we talked about a little bit about the digestion, we talked a little bit about the absorption. Now let's although we will not be able to complete it in this discussion, but let's talk a little bit in this discussion about the glucose metabolism, starting with the first biochemical process pertaining to carbohydrate metabolism, which is glycolysis. Okay, so we talk a little bit about glycolysis. We won't be able to complete it in this discussion, so there will be a discussion coming after this, a video coming after this, that will complete glycolysis as such, but at least let's start glycolysis in this video. So we talk about the actual metabolism. Okay. Now before we go on to the actual metabolism of carbohydrates, it's a little important to know, or rather not little important, it's very important to know one last thing pertaining to glucose as such. That is the normal level of glucose in the blood. Very, very important. Very, very important. It's so important to know the value of glucose. It should rather be on your epitaph, in your grave when you die. Embed it that well in your brain. That well, because there are so many cases, there will be so many cases regarding abnormal glucose levels in blood, right? That's the most common case patients complain of. So here is the thing. The normal, let me write it here in a side note. See, the normal fasting plasma glucose level, the normal fasting plasma glucose level is in the range of 70 to 110 mg per deciliter. Very, very important. We have to give, or rather we have to specify so many parameters at the beginning. It has to be normal fasting because obviously food intake will change the glucose level in blood. Normal fasting, and obviously since the glucose is being extracted from the blood plasma, so plasma glucose level, that is in the range of 70 to 110 mg per deciliter. Okay. Now obviously if we take a heavy carbohydrate meal, then obviously it will go up, but here is the ramification. It should never go beyond 150 mg per deciliter, even in normal, or rather in normal people, a person. It should never go beyond 150 mg per dl even after a rich carbohydrate diet. Okay, so these two values, please remember them, please. Now let's talk a little bit about glycolysis. So we are talking about glycolysis first because obviously it's pertaining to the glucose metabolism. So what is glucose metabolism? Obviously we have to define that first. And so the thing is that glucose has been absorbed by the body, right? Now the body has to do something with glucose. Why did it absorb it in the first place if there is no work to do with it? So obviously glucose has some functions in the body. We're not going into the details of that, but we do know that glucose has these different functions in the body, right? For those functions to be carried out, glucose has to go through a series of different steps, series of different metabolic pathways. These metabolic pathways constitute what we call to be metabolism of glucose. Metabolism of glucose. One, or rather the first biochemical pathway which glucose has to go through in every cell of a living person is this pathway which is known as glycolysis. Okay, glycolysis, very, very important. It's also known as the EMP pathway. EMP standing for Embden Meyerhof and P for Parnas. Okay, so Embden EMBDEN, Embden Meyerhof Parnas pathway. Okay, so these are the name of the scientists who actually deducted the steps of glycolysis. So glycolysis, obviously it's a pathway in which glucose is broken down. So this is a catabolic pathway. Okay, Glycolysis is the catabolic pathway because glucose is being broken down here. What is glucose broken down into? Glucose is a 6 carbon compound. So it will be broken down into 2 3 carbon compounds. And these 3 carbon compounds will in turn be at the very end converted into pyruvate, which is a 3 carbon compound, by the way, right? So this series of steps converting glucose to pyruvate is known as glycolysis. So glycolysis, glyco means sweet and lysis or lysis means splitting. So splitting of glucose, right? So thing is that pyruvate is the end product of glycolysis. There is no doubt about that. But there is a twist here. Twist is that after pyruvate is formed, the fate of pyruvate depends on whether the cell in which glycolysis is going on is getting enough oxygen or not. So under aerobic conditions, when oxygen is plentiful, pyruvate goes on to what we call to be the oxidative decarboxylation. We all know this, right? These are not things that we are decarboxylation. Yeah, so these are not things that we are, maybe listening to for the first time and, oh, okay, so that's that. No, we have all learned about these, I hope so. So this is just a recap of things. So oxidative decarboxylation, it will be converted to acetyl CoA and then it will go on to the very special Krebs cycle, right? But if the conditions are anaerobic, oxygen is not sufficient, then pyruvate will be converted to a special compound known as lactate, okay, by the enzyme the lactate dehydrogenase, right? So anyways, that's the fate of pyruvate, and pyruvate itself is formed as the end product of glycolysis, right? Now, what is the site? Where does glycolysis take place? Firstly, glycolysis takes place in the cytoplasm of cells. Cytoplasm. Which cells? Almost all cells. That's why I said that it is universal. It's a universal phenomenon. It happens in almost all living cells. So almost all cells perform glycolysis. And where in the cells? Cytoplasm. Not the mitochondria, not any other cell organelle, cytoplasm itself. Okay? Okay. Now, the thing is that what is the reason for performing glycolysis? See, we are not jumping into glycolysis as the metabolic pathway itself and just writing down the steps and regulation. No, we'll do that. But first we have to know the basic premises of glycolysis. Why do we need glycolysis? Why does the body actually need to go through the pain of this process? And the reason is very simple. See, why do we need glucose at the first place? We need glucose because we need an energy source. That's the first and the primary reason for glucose, right? We need an energy source for our day-to-day life activities. So glucose is the most preferred energy source, right? But glucose itself directly cannot be utilized for energy. After all, our energy is stored in the form of high energy bonds, right? For example, the bond between adenosine diphosphate and the third phosphate bond, which we call to be ATP, adenosine triphosphate, the energy currency. So we, roughly speaking, we use energy more as ATP. So if we are saying that glucose is the metabolic fuel of the body, then somewhere glucose has to be utilized in such a way that we get that energy currency, ATP, right? So in other words, if I am to say that glucose is the ATM machine, it is the ATM machine, okay? And from the ATM machine of glucose, we are actually taking out the currency or money in the form of ATP, adenosine triphosphate. So glucose is the ATM, adenosine triphosphate is the currency. That's how I look at it. Okay? Now, so that's one reason why glycolysis is significant for our body, or why our body goes on through glycolysis. Glycolysis is one of those, or rather the first preliminary metabolic pathway for glucose by which ATP is manufactured from glucose. Okay? Be it anaerobic conditions, be it aerobic conditions. Okay? So energy is the first significance for glycolysis to actually occur. Now, second thing is that glycolysis, when we will talk about the different steps and when we will talk about the different aspects of carbohydrate metabolism other than glycolysis, gluconeogenesis, then we'll talk about Krebs cycle and such. So then we'll be talking about how the steps of glycolysis are not only important for energy production but also for synthesis of other compounds which in turn are essential for the production of, for example, the non-essential amino acids, glycerol part of fat. All of these are actually synthesized from the glycolytic pathway itself. So glycolysis is actually a series of steps happening and a different number of compounds being connected to each other through metabolic pathway. And these compounds, which are the intermediates of glycolysis, they themselves can go into side reactions and side metabolic pathways, and in turn they will form different compounds which are useful for the body. For example, as I have exemplified, for example, the non-essential amino acids, right, or the glycerol part of fat, or important compounds such as 2,3-bisphosphoglycerate, which are very important for erythrocytes, right? So synthesis of different compounds is another significance of glycolysis. Not to mention the energy source, glycolysis being an energy source, and in fact glycolysis being the only energy source in case of anaerobic conditions or anaerobic cells, because the anaerobic cells also need energy, but for energy to be there, for ATP to be manufactured, the cell has to go through different biochemical pathways. For example, the Krebs cycle, the electron transport chain, different kinds of for example oxidative phosphorylation, right? So but all of them have the prerequisite of requiring oxygen. Anaerobic cells do not have oxygen, or rather sufficient oxygen. So glycolysis for the anaerobic cells is the only source of energy where oxygen is not required, but they get the bare minimum for sustaining themselves. Okay, done. And by the way, glycolysis is the breakdown of glucose. So you're breaking down glucose into something. And what is the manufacturing of glucose from something else? Called gluconeogenesis. So glycolysis and gluconeogenesis, they are reciprocal. They're reciprocal methods. Glycolysis is breaking down, gluconeogenesis is building up of glucose. So what's seen is that, why I'm talking about this is that glycolysis is also important for gluconeogenesis to take place. As we'll see in glycolysis, we'll see that glycolysis, the steps of glycolysis, mostly they are reversible. Some irreversible steps are also present, but most steps of glycolysis are reversible. So gluconeogenesis will actually follow the reversible pathway, and it will circumvent the irreversible steps, right? It will follow the reversible steps. Gluconeogenesis, it will follow the reversible steps. It will circumvent the irreversible steps somehow. We'll talk about that later, and it will form glucose at the end. So the framework of glycolysis is important and significant for gluconeogenesis also. Okay, done. So having said this, we now can actually discuss the different steps of glycolysis. We can now talk about the different steps of glycolysis. Okay, so let's quickly lay down the glycolysis pathway. Let's talk about it in a different one. Now, as far as remembering the glycolysis pathway, I don't have any memory aid because I learned it the hard way, which is complete memorization. There is no other way to remember glycolysis. But the good thing is that glycolysis is taught in schools also, in the 11th and 12th standards, the 11th standard. So at least in Indian schools they are. So that's why that basic backing up is there. Anyway, so if you don't know the different the steps of glycolysis, then please do remember them. Please do memorize them. There is nothing I can do here for you. You have to remember it. If you can find a memory aid somewhere on the internet, very well, amazing. But let's get on with it then. So at the very first, obviously we have glucose. No problem with that. Glucose is converted to glucose 6-phosphate. Glucose 6-phosphate means that in the 6th carbon of glucose chain, the 6th carbon of the glucose carbon skeleton, a phosphate is added. We'll add the enzymes and the different aspects later. Let's first draw the basic skeleton. So glucose 6-phosphate, done. Now glucose 6-phosphate is converted into fructose 6-phosphate. Okay, done. Fructose 6-phosphate is in turn converted into fructose 1,6-bisphosphate. That too done. Okay, fructose 1,6-bisphosphate is converted into, or rather split into, two compounds. Very important. This is where the splitting takes place. Up to now the compounds were 6 carbon. Now they split into 3 carbon compounds. What are they? Fructose 1,6-bisphosphate will break down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, these are the two compounds. Now dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they are readily interconvertible. Okay, and what happens is that glyceraldehyde 3-phosphate will in turn be converted into what we call to be 1,3-bisphosphoglycerate. Okay, 1,3-bisphosphoglycerate will in turn be converted into what we call to be the 3-phosphoglycerate. Done. 3-phosphoglycerate will be converted into 2-phosphoglycerate. phosphoglycerate. Okay, 2-phosphoglycerate will be converted in turn into what we call to be phosphoenol pyruvate. Phosphoenol pyruvate will be converted into pyruvate, and pyruvate is the end product of glycolysis. However, if this is an anaerobic condition, then pyruvate in turn will also be converted into lactate. Okay, so this is the basic skeleton of glycolysis, which I really do not have any memory aid to help you with to memorize this. Okay, I have memorized this the hard way again, I'm telling this, but it's good that my biology teacher was very strict, and so I had to learn it anyways. So that's helping me out right now, right? Anyways, so now let's fill in the blanks. We have already drawn the basic framework, we have drawn the basic skeleton. Now let's fill in the arrows with the enzymes and the different aspects. So the first reaction, glucose to glucose 6-phosphate, is getting a phosphate, right? It was glucose, then suddenly there is a phosphate. Where is the phosphate coming from? Yes, it is coming from ATP. So the first step will require ATP to ADP, right? What is the name of the enzyme which is facilitating? The name of the enzyme is, now this is important, and we'll talk about this in the next discussion. The enzyme which facilitates glucose to glucose 6-phosphate is a hexokinase or an isozyme of hexokinase called glucokinase. Okay, so hexokinase or glucokinase, these are actually two isozymes, like they are isozymes of each other, isoenzymes of each other, isozymic forms of each other. We will talk about the differences between these two enzymes in the next discussion, but for now just remember that hexokinase or glucokinase, they are isozymes. Either one of them will facilitate glucose to glucose 6-phosphate conversion. And by the way, this enzyme will use magnesium as a cofactor. So magnesium ion is used as a cofactor by this enzyme. Done. Glucose 6-phosphate to fructose 6-phosphate is converted by phosphohexose isomerase or phosphoglucoisomerase. Okay, so I have seen different books print out different things. Phosphoglucoisomerase, or I have also seen phosphohexose isomerase. Phosphohexose isomerase. Right. And obviously isomerase, the name is justified. Glucose 6-phosphate to fructose 6-phosphate, and glucose and fructose are isomers of each other. So, and the 6-phosphate condition remains the same. So glucose to fructose isomerization is taking place, so an isomerase. And phospho, since there is a phosphate here, gluco, since it's gluco, or hexose, because these are hexose sugars. So there is a justification behind the name of the enzymes, right? For example, if I am talking about kinase, a kinase, it's a family of enzymes, it's a class of enzymes, and kinase enzymes, actually what they do is that they phosphorylate, they help in phosphorylation. So that's why glucose to glucose 6-phosphate phosphorylation is taking place with the help of hexo, because glucose is a hexose sugar, hexokinase or glucokinase. Just like that, glucose 6-phosphate to fructose 6-phosphate, the isomerization is taking place with the help of phosphoglucoisomerase or phosphohexose isomerase. Done. Then the next step is again an irreversible step. So the first step is an irreversible step. The third step is also an irreversible step. Okay, fructose 6-phosphate to fructose 1,6-bisphosphate, right? So again we have a phosphorylation taking place. So the name of the enzyme is another kinase, right? The name of the enzyme here is phosphofructokinase or PFK. Phosphofructokinase. Now, as you can see, I have not closed the bracket here. Why I have not closed the bracket is because phosphofructokinase, this enzyme has two forms. It has two forms. We'll talk about this later in the next discussion in details. Two forms, right? One is called PFK1 and the other is called PFK2. Okay? The enzyme, the isozyme, so PFK1 and PFK2, these are isozymes of each other, right? Fructose 6-phosphate to fructose 1,6-bisphosphate. This reaction is carried out by the enzyme PFK1. And PFK2's action we'll see in the next discussion. It's not directly related to glycolysis. Indirectly related to the regulation of glycolysis, yes, later. Okay. So phosphofructokinase 1, this is the enzyme. And hence obviously we need a phosphate source, so an ATP is hydrolyzed, ATP to ADP. And by the way, since it's a kinase, just like hexokinase here or glucokinase, whatever, it is also requiring a magnesium as cofactor. In fact, most of the enzymes in the glycolysis require magnesium as cofactor. Okay. And the reason for using magnesium as a cofactor, there are a lot of reasons, but one of the major reasons is that magnesium is forming a complex with the enzymes, which is lowering the activation energy of the particular step. That is one of the reasons why magnesium is used as a cofactor in many of these enzymes. So magnesium actually forms a complex with the enzyme, which in turn will obviously bind with the substrate, leading to lowering of the activation energy of the specified step, and hence easily the step will occur. Okay, so that's just a small information. So now fructose 1,6-bisphosphate will be converted, or rather will be split to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. Okay, aldolase. Again, just like phosphofructokinase, aldolase also has two isozymes. In fact, up to now, all the enzymes that we have studied, they had two isozymes, right? Aldolase also has two isozymes. We'll not talk about this now. It has two forms, aldolase A and aldolase B, not aldolase 1 and aldolase 2. In fact, if I am to remember correctly, aldolase A and aldolase B are not the only isozyme forms of aldolase. There are a lot of other isozyme forms also, but A and B are the most common ones in case of aldolase. Anyways, we'll talk about that later. So that's aldolase, right? We're only laying down the basic foundation for our next discussion of glycolysis. Then we talk about glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now see, all the glycolysis steps from here carry on with glyceraldehyde 3-phosphate as a substrate, not with dihydroxyacetone phosphate. So what happens is that dihydroxyacetone phosphate will be converted to glyceraldehyde 3-phosphate. So we will have two molecules of glyceraldehyde 3-phosphate. One molecule is coming directly from fructose 1,6-bisphosphate. The other is coming from dihydroxyacetone phosphate's isomerization. So this again is also another isomerase enzyme. In fact, it's known as phosphotriose isomerase. Just like the phosphohexose isomerase, since the phosphohexose isomerase was dealing with hexose sugars, now we have phosphotriose isomerase because we are dealing with triose or 3 carbon sugars, Glyceryl dihydrophosphate and dihydroxyacetone phosphate. So the name of the enzyme is phosphotriose isomerase. Done. Now the next step is very important. Why it's very important? It's very important for a number of reasons. Number one reason is that, first let's write the name of the enzyme here. See, glyceryl dihydrophosphate 3-phosphate is being converted to 1,3-bisphosphoglycerate, but there is a but. Although phosphorylation of glyceryl dihydrophosphate 3-phosphate is taking place here, but phosphorylation is not the only thing taking place here. What is the other thing taking place in this reaction? The other thing is that there is a dehydrogenation also going on. NAD plus to NADH plus H plus. Very, very important. This is the first reason why this step is so important. This is the first reason of the importance of this step. There are other reasons also, I'm coming to them. NAD plus to NADH plus H plus. So we find essentially that this oxidized coenzyme NAD plus, it is reduced to its reduced form NADH plus H plus. So in the actual reaction, which is glyceryl dihydrophosphate 3-phosphate to 1,3-bisphosphoglycerate, what is taking place? Yes, dehydrogenation is taking place because the hydrogen from glyceryl dihydrophosphate 3-phosphate is going to NAD plus. So dehydrogenation is taking place. So the enzyme required will be a dehydrogenase enzyme. The name of the enzyme is the name of the substrate plus dehydrogenase. That is glyceryl dihydrophosphate 3-phosphate, just like the name of the substrate, dehydrogenase. Glyceryl dihydrophosphate 3-phosphate dehydrogenase. So glyceryl dihydrophosphate 3-phosphate dehydrogenase is the name of the enzyme, and obviously that's pretty logical, right? So we obviously need glyceryl dihydrophosphate 3-phosphate to 1,3-bisphosphoglycerate. We need a phosphate, right? Now for the other reactions where phosphate was needed, the phosphate was provided by ATP. But in this case, the phosphate will not be provided by ATP, but in the form of an inorganic phosphate itself. So it will not come in the form of a particular compound. Like for example, a particular compound, example ATP. ATP is a compound. It arrived, gave its phosphate, turned into ADP and left. No. In this reaction, the inorganic phosphate will be added itself. Okay? No other source. Direct. Now, 1,3-bisphosphoglycerate formed, right? 1,3-bisphosphoglycerate after being formed is converted into 3-phosphoglycerate, right? So 1,3-bisphosphoglycerate to 3-phosphoglycerate will take place by 1,3-bisphosphoglycerate kinase. 1,3-bisphosphoglycerate kinase. And this step will be the first step in the glycolysis reaction or glycolysis pathway, where an ATP will be formed. Rather than an ATP being consumed, a molecule of ATP will be formed from this step. So it's very, very important. And guess what? Just like any other kinase of the reaction, hexokinase or this phosphofructokinase, 1,3-bisphosphoglycerate kinase also requires magnesium ion as cofactor. So that's pretty good, right? That's a memory aid itself. So 1,3-bisphosphoglycerate kinase will lead to the formation of 1,3-bisphosphoglycerate to 3-phosphoglycerate. Now the name of this enzyme is a kinase, but we are not seeing a phosphorylation, we are seeing a dephosphorylation. Why? Because this reaction is reversible. So in terms of the way in which we are seeing, that is 1,3-bis phosphoglycerate, 2,3-phosphoglycerate, maybe it's not a phosphorylation, but obviously it's a reversible reaction. So we can see the other way also, 3-phosphoglycerate to 1,3-bis phosphoglycerate. If we see it the other way, we will find that 1,3-bis phosphoglycerate kinase, the name is justified. In the other cases where kinases were required, they were irreversible steps. Hexokinase or glucokinase. There was only one way to go, glucose to glucose 6-phosphate, not the other way around. Here also fructose 6-phosphate to fructose 1,6-bis phosphate, not the other way around. Right? Although these steps can be circumvented in gluconeogenesis, we'll talk about that later. Anyways, so this is one such step where 1,3-bis phosphoglycerate kinase, the name is justified because it's reversible. Okay? And obviously an ATP is formed. Then from 3-phosphoglycerate to 2-phosphoglycerate, the enzyme required is mutase, that is phosphoglyceromutase. See, this is not an isomerase but a mutase enzyme. What that means is that 3-phosphoglycerate and 2-phosphoglycerate are not isomers as such, right? They differ in the phosphate arrangement. So it's not a phosphoglyceroisomerase but a phosphoglyceromutase, which will just change the configuration or the position of the phosphate, change it from the 3rd carbon to the 2nd carbon. So 2-phosphoglycerate is formed. The next step, 2-phosphoglycerate to phosphoenol pyruvate, is another important step, because this is one such step where the enzyme is obviously enolase, and here dehydration takes place. So one molecule of water is given out. One molecule of water is given out. Done. So 2-phosphoglycerate to phosphoenol pyruvate, enolase is the enzyme, very important, and obviously, and this will require magnesium. This is not a kinase enzyme, but it will require a magnesium. Okay. So enolase is the enzyme to convert 2-phosphoglycerate to phosphoenol pyruvate, and a molecule of water is actually given out. Last step is phosphoenol pyruvate to pyruvate, and the reaction is catalyzed by pyruvate kinase enzyme. Pyruvate kinase enzyme. Okay, and this is another one of those steps where ATP is formed, ADP to ATP, and being a kinase, it will require magnesium. Done. So that will complete the basic. We only wrote the enzymes, the different substrates, the different the actual carbon skeleton. Not the carbon skeleton, the skeleton of the metabolic pathway. So we completed the actual thing. And last but not the least, pyruvate to lactate, the enzyme is lactate dehydrogenase. Very important enzyme, lactate dehydrogenase. So what will happen in this reaction is that we had seen that NAD plus is being converted to NADH plus H plus. In this enzyme, the reverse will take place. That is, the formed NADH plus H plus, let me write it here, NADH plus H plus, the formed, I'm sorry, it's becoming a little messy, that will be converted to NAD plus. And that NAD plus will again be reutilized here. Right? Again, it will be reutilized over here. Done. So that completes, I would say, just the basic fundamental framework for glycolysis. The basic foundation for glycolysis has been laid. Now when we come back the next time, we will be talking about the details, the specific steps of glycolysis in a little bit more detail, its regulation, and we'll also complete our next discussion with the fructose and galactose metabolism. There are just one or two different pathways for those two, not to worry. And with that, we'll complete the next discussion. So these two discussions will together complete the first part of carbohydrate metabolism. So thank you very much. Hope you got something from this entire discussion and it was not entirely a time waste. So thank you. I'll see you later. Thanks.